In this video, we go over the innervation of our muscles, joints, and skin. We'll start off with the motor components, so innervation of the muscle. We'll then move into the sensory components that innervate the muscle. And finally, we'll talk about the afferents that innervate the skin. The skin afferents are going to sense pressure for tactile sensation, so anything that's not painful. And then pain arises whenever we have changes in pH or temperature. So the first two parts are about controlling the muscles. And really controlling our muscles is the most important task of the nervous system. That's what moves our body around in space and allows us to navigate the world. So the first concept that we need to go through is that of the motor unit. And a motor unit is just a motor neuron and all the muscle fibers that it innervates. Motor units come in different sizes depending on the fineness of movement required and the amount of force that's required. So in general, the finer the movements and the less force that's required, the smaller the motor unit. So one neuron will control relatively few muscle fibers. That may be just a handful. Larger motor units arise whenever we need to generate more power. We don't necessarily need a whole lot of fine movements, like in the back, for example. Larger motor units there than, for example, in the eyes. The recruitment of motor units occurs in an orderly fashion. This is described by what we call the size principle. In general, the smaller and therefore weaker motor units are recruited first, and then the larger, more powerful motor units are recruited later. This way we get smooth, gradual activation of muscles and not jerky movements. So first, just a bit of review on muscle cell structure and the physiology of muscle contraction. So the basic building block of muscles are sarcomeres. And the sarcomere is really just the collection of thin actin and thick myosin filaments that lie between the Z discs or Z bands. So the sliding filament model for muscle contraction. Here's our Z band here. Actin fibers coming off here, and the actin in our muscle is much more stable than actin elsewhere. So this is a heavily modified version of actin that really doesn't change its size. So we have fairly stable actin filaments, and then we have our myosin thick filaments. Myosin is still a motor protein. There's a bunch of these myosin heads here, so that's the little hairs that I'm drawing off here. Myosin pulls on actin. And so we have motor proteins oriented in different directions, so that we'll pull the actin in different directions. Okay, got a little sloppy at the end. But muscle contraction involves the shortening of sarcomeres by sliding these filaments along each other. So we end up with a shorter distance between our Z bands, but the actual filaments are the same length. Nothing's really changed its size here. It's just we've gotten rid of that space in the middle between our actin filaments. The thick myosin fibers are exactly the same size. The thin actin filaments are exactly the same size. What happened was the myosin motor protein pulled on the actin filaments. Okay. Sometimes you gotta let it go. So we have sliding filaments. Slide together. So the sarcomeres change their size, and since muscles are just made of a whole bunch of kind of sarcomeres lined up in a row, as each one shrinks just a little bit because of sliding filaments, the whole muscle can have an overt change in its size. So muscles are these syncytial cells where a whole bunch of individual cells kind of fuse their cytoplasm. And that's why each muscle cell has many different nuclei located around the surface, because many cells fused together to create one single, very, very large muscle cell. So the muscle cells are then made of these repeating sarcomeres. So there'll be a sarcomere here and there and over there and all along the wall there. So my muscle, from one end to the other, a whole bunch of sarcomeres lined up. All the individual strands, those are single cells, which of course are made of many cells fused together. So we got actin filaments, those are the same old cytoskeletal component, just a little more stable. And then we got the myosin filaments. Those are a little thicker, they have the motor protein myosin on them. So they're going to burn a little ATP to pull on the actin filaments. And we're pulling in different directions on both ends toward the center, so our filaments slide in. So here you can see there in that cartoon, the sarcomeres are shortening in order to change the length of muscle fibers. So the thick pink myosin filament there, it's got the little myosin heads that are going to swim along the actin filaments, pulling those inward toward the center. And that will cause the shortening of our sarcomere. Shortening the sarcomere shortens the muscle. The power is generated by myosin. And there's a couple different types of myosin, but they're all going to do the same thing. They're going to burn ATP in order to pull actin somewhere around 10 nanometers or so for every ATP that's burned. So it's probably worth knowing this cycle, since this is how muscles work. And knowing how muscles work is probably important to you. So we've got our thick filament here, and we'll just do one myosin head. This will bind to the actin filaments. So we got our thick myofibril here, which contains myosin, the motor protein. Myosin is an ATPase, meaning it's an enzyme that burns ATP. It's going to bind ATP, break it down to ADP, and this is going to drive some sort of change in shape. Just like the sodium-potassium ATPase burns ATP to move sodium and potassium, myosin burns ATP to pull on actin. So, the binding between myosin and actin is going to kick off this little ADP molecule. So resting myosin is bound to ADP. Remember, it's an ATPase, so it burns, it breaks down ATP. The binding event that occurs here, let's just imagine this over time, it's going to destabilize the interaction between myosin and ADP. So myosin can only hold one thing at a time. So when it binds to actin, it has to let go of ADP. When it lets go of ADP, that causes a change in its shape. Instead of being forward, it cocks back. So let's imagine our filament now. So we'll just kind of go across time here. So this is a big cycle. I'm recreating the drawing on the slide for you with a little bit of narration. So we got our thin actin filament, and its job is to just sit there, don't move, just get pulled on by myosin. When we release ADP, that causes myosin to change its shape. Right? It used to be over here, and then it kicks back. That pulls on actin about 10 nanometers, so a very small distance. So we're going to need many of these cycles to pull on our actin fibers, but it gives us very fine control over the length of the muscle. Now that we've let go of ADP, the binding site for the adenosine nucleotide has opened up. It's not going to bind to ADP. All right, it's already holding on to actin. The only thing that it prefers more than actin is ATP. When that occurs, again, myosin can only hold on to one thing, so it lets go of actin. Myosin is an ATPase, so it spontaneously breaks down ATP to form ADP. When it's ADP bound, only then is the myosin head forward. So now we're back where we were. So here's the preference for myosin binding. This is what myosin prefers. Above all else, ATP. The problem, though, is that myosin is an ATPase, so it breaks it down to ADP. Maybe you're familiar with that, there's a cartoon, I'll hug him and squeeze him and call him George, and there's a, I think it's a cat maybe, and it just hugs stuff too much and kills it. So that's what goes on here. Myosin loves ATP so much. It wants to bind it above all else, but it's an ATPase. It eventually breaks it down. It hugs it and squeezes it. Rather than calling it George, it hydrolyzes it into ADP. In between these two, we have actin. That's my preference. If I'm myosin, ATP above all else. If I don't have that, I'll take actin. And I guess I can hold on to ADP if there's no actin around. I'm going to hold something. The question is what? So, when it hydrolyzes the ATP to ADP, it prefers to bind actin. When it binds actin, it lets go of ADP, so now it can bind ATP again. And so we have this cycle here. When myosin is allowed to interact with actin, it binds, lets go of ADP. When it lets go of ADP, it changes its shape, pulling on actin. Once ADP is out of the picture, ATP can bind. It can only hold on to one thing, so it lets go of actin to hold on to ATP. It hugs it, squeezes it, and then we have ADP. Then we can bind again, let go, pull, bind ATP, and the whole cycle continues. And that's what's going to drive the shortening of sarcomeres. So muscular contraction occurs because of the hydrolysis of ATP by myosin. It's going to drive changes in its shape. So this myosin power stroke here. So we have our cycle. So in here, the first thing we do, we start off kind of here. Step one, we're going to bind. We can only hold on to one thing, right? So we got to let go of ADP. When we do that, that changes the shape. So myosin kicks back. Myosin prefers to bind ATP, and there is ATP floating around. So when it binds ATP, it lets go of actin. That breaks that cross bridge between myosin and actin. Myosin is an ATPase, so it will eventually break down ATP into ADP and cock it forward again so the whole thing can be restarted. So in that forward conformation, that's ADP bound myosin. When it lets go of ADP, it pulls back. When it has ADP, it's forward. Now, actin and myosin interactions are only allowed whenever there's sufficient calcium levels within the muscle. So the muscle has to be active, in other words. If the muscle is not active, actin-myosin interactions are allowed because that binding site is covered by tropomyosin. So the reason why this is sitting here, just waiting, is because we have a protein in the way. We got this, well, maybe it's more like a helix kind of wrapping around it. We've got tropomyosin. Wraps around actin and obscures the myosin binding site. Tropomyosin is pulled out of the way by the calcium binding protein, troponin. So when we have a burst of calcium, because of input from the motor neuron, as we'll find out, that calcium binds to troponin, and troponin then pulls tropomyosin out of the way. That might be a little easier to see in cartoon format. So, there tropomyosin is green in this case, troponin, little blue balls there. That's the calcium binding protein. When it binds calcium, it changes its shape. So when it changes its shape, any protein that it's attached to gets pulled. So once we have calcium release within the muscle, troponin pulls tropomyosin out of the way, revealing the myosin binding site on actin, so myosin can bind. When it binds, it spits out to ADP, changes shape. And that whole thing happens. Now, different muscle fibers are going to have different contractility, different degrees of force that they can generate, because not every myosin head is created the same. Some muscles have myosin heads that hydrolyze ATP fairly slowly, and so they contract slowly and generate very little force. Now the good thing about them, because they hydrolyze ATP so slowly, and because they have a bias toward aerobic metabolism, they don't fatigue very often. So a slow, I think I can get rid of this. I'll probably regret it, but we'll see. So there's a couple things to think about with these different types of fibers. So there's for each fiber we need to think about the force that it generates, and this depends on myosin. How quickly does myosin hydrolyze ATP? And then we need to think about the type of metabolism. I guess if I wanted all F's, feeding might have made sense to put there. So the first type of fiber, type 1, slow twitch. So very slow. So we're not burning a lot of ATP, we're not generating a big bill, and we primarily use aerobic metabolism, so we make a whole lot of ATP. So because we're slow, very little ATP used, but a whole lot of ATP made. And that's why these don't fatigue. So slow twitch fibers, although they don't generate a lot of force, they'll work all day. So your postural muscles, your respiratory muscles, a lot of slow twitch in there. And this changes over time. Because we breathe all day, every day, the respiratory muscles are going to include a whole lot more type 1 fibers as we age. So newborns will have a little more fast twitch there. But then as we age, we're going to see more slow twitch come on. So these things are plastic. The amount of fibers change. Now, when we get to type 2, there's really two types of fast twitch fibers. So both of these are fast. Maybe a little bit faster here, but both fast. You know what, I'll say very fast. Why not? Why not? So as we increase the speed of myosin, we increase the ATP used. Unfortunately, we also see a change in their preference for metabolism. So we see a mix of anaerobic and aerobic. So glycolysis is anaerobic, and then the oxidative phosphorylation aerobic. And then there's a heavier bias towards glycolysis. You see a lot more glycolytic enzymes in the type 2B. So even though they're running up a big bill, they're not generating so much ATP as our slower twitch fibers. And we can see those three types of fibers over here in this illustration. So the star showing you type 1. So the alkaline labile myosin, this is the faster version of myosin. You don't really see a lot of staining in the type 1 fiber, shown kind of on the left there. You see a moderate amount here, that's why it's just fast, not very fast, and you see a much heavier amount in the type 2B, so in the circle there on the top left. So that alkaline myosin ATPase, that can help us distinguish these types of fibers. How much of the fast myosin ATPase do you have? And then we can look at the presence of glycolytic enzymes. So do we have a lot of glycolytic enzymes around? Well, we don't see a ton of them in the type 1 fiber. Again, they're a little more biased toward aerobic. Of course, they have some. You have to go through glycolysis in order to go through aerobic metabolism. We see a lot more of them over here. When we look at NADH, that's an electron carrier that's critical for aerobic metabolism. Again, you're seeing more here in type 1 and type 2A. You don't see a whole lot in the type 2B fiber because they're primed more for glycolysis. So you get this difference in the amount of force you can generate and the amount of ATP that you generate, and you see an inverse relationship. The more force you're generating, the fewer ATP molecules you're able to generate over time, and that's why those strong muscles fatigue. Now, depending on usage, the makeup of muscles can change with time. So again, after we're born, we breathe, and we continue to breathe the rest of our lives. It's probably the last thing we do before we die. And because we're using those muscles all the time, you see a shift towards slow twitch fibers. We can also see that here in these data. So they hooked up a nerve stimulator to provide very weak stimulus in the one of those leg muscles. What's that say? Medial gastrocnemius or something like that? I don't know. Don't judge me. At the start of the experiment, when they deliver strong stimulation, the amount of force that they get is a whole lot greater than after their experiment. So the top there is just giving you a cartoon of what they're doing. So they're providing low levels of stimulation to the nerve. So force over time. Now with faster twitch fibers, you generate a lot of force quickly. With that low level of stimulation, where we're really only getting the kind of smaller, weaker type 1 fibers, over time what they saw was the force generation curve looked a whole lot different. With their low level of stimulation, they saw an increase in type 1 fibers, because those are the muscles that were being used more than the others, so use it or lose it. So the makeup of muscles can change over time with use or disuse, I suppose. And the type of muscle that's innervated relates to the type of motor neuron that innervates it. So what's quite, I think, surprising is that different types of muscle fibers tend to be targeted by different sized motor neurons. So we have a relationship between motor neuron and muscle where the smaller motor neurons tend to innervate the type S fibers, whereas the type 2 fibers are going to be innervated by slightly larger motor neurons. So when we consider motor units, the motor neuron and all the muscle fibers it innervates, we get, it's gone, we get the same kind of 3 levels. But instead of being type 1, type 2A, and type 2B, we call them names that I think make a little more sense. Type S, slow twitch. You can't fatigue these things. These will work basically all day. Fatigue resistant. These are those fast but fatigue resistant, so type 2A. And then fast fatigable, again, I guess very fast, I suppose. Type 2B fibers. So we have 3 different types of motor units. And the motor unit, again, is just the motor neuron and all the muscle fibers that it innervates. And motor neurons tend to target the same type of muscle fiber. FF gives you a lot of force, but it fatigues quickly. Type S gives you very little force, but it'll give you very little force all day, and sometimes that's all you need. Now the neurons that innervate these different types of motor units that says motor unit come in different sizes, and that's what creates the size principle. So those neurons that target the slow or the type S motor units, a little smaller motor neuron compared to the fatigue resistant and the fast fatigable. And the size matters here because the size determines the internal resistance of the cell. And I'm sure you imagined that this was coming. V equals IR, Ohm's law. That hasn't changed. So the amount of potential that we get from excitatory currents depends on our internal resistance. So the smaller neurons are going to hit threshold potential first because they have a higher internal resistance than the larger ones. So given the same current, you get a much larger potential in the small ones. What this means for us is that our weaker muscles get recruited first. So that whenever we initiate a movement, we're not generating maximal force, we're generating just a little bit of force. If we encounter resistance, we then recruit larger motor units to overcome that resistance. But we don't use those fast fatigable fibers because we don't need them unless there's some resistance. And they burn up a lot of ATP but don't make it. So we don't want to use them unless we need them. So the first thing we recruit are the type S motor units. And the reason we do that is because of Ohm's law. We have smaller neurons, so they're a little more excitable. And that's what the cartoon is showing us there. The smaller your neuron, the more excitable it is for a given current. The interface between motor neurons and the muscle fibers is still the neuromuscular junction. And this is still a massive synapse, has about 1,000 active sites. Each one of these is going to generate about 1 millimolar concentrations of acetylcholine in the synapse, something like that. If we look on the postsynaptic side of things, sorry, I'm not good with words, good thing I'm talking. We see those postjunctional folds. We can see them over here, low mag on top, little higher mag on the bottom. We know the top is the presynaptic terminal because it has those vesicles in it. And on the postsynaptic side of things, we have postjunctional folds to increase the surface area. Right, there's multiple active zones. We're going to increase the surface area for neurotransmitter receptors, and then we'll also trap neurotransmitters if they try to diffuse around. And of course they're going to diffuse. They could leave, or they could travel into these postjunctional folds so that they continue to remain at the synapse and continue to depolarize the muscle just a little bit. At these postsynaptic sites, we find nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors, and these are still non-specific cation channels. So they reverse around 0 millivolts, which is well above the threshold needed to generate an action potential in the muscle. So when we release acetylcholine from a motor neuron, sometimes we just put it together in one word, motor neuron. They release acetylcholine. That stimulates nicotinic acetylcholine receptors, and we depolarize the muscle. Cool. But of course we want to change the shape of the muscle. We want to change the length of the muscle, not just the membrane potential. But in order to change the shape, we have to somehow liberate calcium to bind to troponin and move tropomyosin out of the way. Well, the easiest way to do that would be through calcium channels. And as luck would have it, the muscle cell is full of voltage-gated calcium channels. Kind of a special kind of voltage-gated calcium channel. They're called dihydropyridine receptors, in case you care, but you don't. Doesn't matter. These voltage-gated calcium channels are intimately connected with ryanodine receptors. So think of these as forming a complex. So whenever we have depolarization from nicotinic acetylcholine receptors, right, that's what's going on here, because it's a non-specific cation channel and reverses at 0 millivolts, it depolarizes us. That depolarization activates voltage-gated calcium channels, which activate ryanodine receptors on the sarcoplasmic reticulum. That's the endoplasmic reticulum for muscles, basically. So it's a big sack of calcium. So when we have input from motor neurons, nicotinic acetylcholine receptors depolarize us, voltage-gated calcium channels are activated by that depolarization, and then ryanodine receptors allow for calcium release from the sarcoplasmic reticulum. And that calcium is critical for changing the shape of the muscle. And that's really what all these data are showing us here. I think you get the idea. Rather than walking through it in too much, let's just look at the blue and green lines there. The blue line is showing you the amount of calcium within the cell. So as the blue line goes up, we have more calcium. As the blue line goes up, that green line on the bottom goes down. That is the muscle length. Increase in calcium, decreased muscle length, because we allowed for contraction and the shortening of our sarcomeres. Rather than me draw all that here, I've made a cartoon for you. So we have input from a motor neuron. That's going to be acetylcholine. That stimulates nicotinic acetylcholine receptors that cause depolarization. Depolarization stimulates that orange voltage-gated calcium channel. The voltage-gated calcium channel then stimulates ryanodine receptors that allow calcium release. Once we have calcium release, then troponin binds calcium, moves tropomyosin out of the way, and we have our actin-myosin interactions, myosin power stroke, sarcomeres shorten. It's pretty nifty. Now, it wouldn't be very helpful, though, if we only depolarize the muscle right here and here. But we don't. Of course, we do depolarize the whole muscle. The whole muscle cell is going to depolarize because it fires an action potential. Now, rather than having an axon that propagates the charge like a neuron does, muscles have transverse tubules. Transverse tubules are lined with voltage-gated calcium and sodium channels to propagate the muscle action potential. So this muscle cell is going to have little perforations in it that'll travel down. So here's my transverse tubule. And all along here are my voltage-gated ion channels, so that when the muscle fires an action potential, it spreads throughout the entire muscle, not just at the surface. So all throughout the muscle, we get synchronized contraction because we have synchronized calcium release. So voltage-gated sodium and voltage-gated calcium channels line the T tubules to spread depolarization. So while it might start at the nicotinic acetylcholine receptors, it's going to spread throughout the muscle by voltage-gated sodium and calcium channels. So they also play a very important role in that depolarization. So the whole muscle contracts. Kind of nifty. And we also have sensory components in the muscle that are going to sense stretch and give us some idea of what's the length of the muscle, both static length and dynamic. So what is the steady state muscle length, and then is it currently changing? We also have stretch receptors within the tendons to sense tension on the muscle. These are going to give us some idea of body position. So if you know the length of every muscle in your body, you could recreate your body. So that's basically what goes on in our mental image of body position. As we change the length of our muscles, we can create an idea of where our body parts are without seeing them, because we sense the length of our muscles. So within the muscle, the sensory component there is the muscle spindle. And the muscle spindle is very important, because this is what creates stretch reflexes. Stretch reflexes, although we call them deep tendon reflexes, aren't really initiated at the tendon. They're initiated within the muscle. We hit a tendon to stretch the muscle. We're not stimulating sensory afferents in the tendon, we're stimulating them in the muscle by stretching it. We do have sensory afferents within the tendon, and this is how I draw muscles and tendons, because I don't really know what they look like. So a big old bag of muscle fibers here. We got some connective tissue as the tendon. Luckily, the name Golgi tendon organ has tendon built right in it, so that's kind of cool. So we have axons kind of woven around the collagen fibers there. That's your Golgi tendon organ. And that'll sense tension. That's its job. Is there too much tension on the muscle where we're running the risk of damaging the connective tissue? If so, inhibit the muscle. Within the muscle, we have little muscle spindles scattered throughout, and these are fairly small. So why don't we blow it up a little bit? There's kind of a central sensory portion. And then there are little tiny muscle fibers on each end. And let's get some sarcomeres on there. There you go. Now we're a muscle. So this is the blow up of the muscle spindle. They're fairly small, so about a centimeter or so, and they're scattered throughout muscles. All right, you'll get to know these quite well as we continue to talk about them. So these sense muscle length, and that's what this central portion does here. That's going to sense muscle length, and the flanking intrafusal muscle fibers are going to let us lie about muscle length, so we can actually change how long our muscles are. The big hunk of meat up here, which used to be my nickname, is made of extrafusal muscle fibers. So all the big muscle fibers that you can see, extrafusal. The little tiny ones that flank the spindle, intrafusal. And different motor neurons target those. The alpha lower motor neurons, which are a little bit larger, hit the extrafusal fibers, so that's what we just talked about. The gamma, little smaller lower motor neurons, hit the intrafusal fibers. There are two types of afferents within the muscle spindles. So the type 1A, a little bit larger, conduct signals a little bit faster, also rapid adapting. These sense a change in muscle length while it's happening. So as it's stretching, they're very stimulated. Once the stretch stops, not as stimulated. So we got two types. There's the 1A that kind of wraps around, if you will. So as this stretches, as the muscle spindle gets stretched because the muscle is stretched, that stretches the axon and pulls open stretch receptors. So we got type 1, and that's the sense changes in length as they're occurring. Then we have some type 2. They kind of splay out. I guess it somehow looks like a flower. I don't know. They call them the flower splaying fibers. I don't know. I do know that they're a little bit smaller, so that's why they're type 2 there. And they're also non-adapting. So they don't have this initial period of high, fast firing followed by slow firing. So they don't tell us the change in length, they tell us the length that it is. How long is it? That's what they're going to tell us. How much stretch is there here? They'll give us some idea of the length. All right. Let's see adaptation. That might help, in case we haven't heard of adaptation before. That's where the firing rate changes. It slows down over time. And that's what these data are showing us here. So on the top, we're looking at the firing rate. On the bottom, it's showing us different degrees of muscle tension or stretch. All right, a little bit of stretch, even more stretch, even more stretch. And this is just no change. And then they stretch at different amounts. With that stretch, they get a big increase in type 1A firing, and then the firing rate drops down. even though the muscle is stretched just as much here and here, the firing, or the frequency of my type 1A activity is not the same. The type 1A fibers are most active during the initial stretch. As that stretch is maintained, they decrease their firing rate. That's what I mean when I say adapting, rapid adapting. So they really only care during the actual change in muscle length. And here it is in cartoon format. So we got our 1A firing rate on the not the very bottom, the one above the very bottom. And on the very bottom, those red ticks are showing us action potentials from the type 2. So those that sense static muscle length. So they're applying some stretch to the muscle. That's what this is showing you here. Give it a little stretch and then remove it. The type 1A shows a whole lot of activity during the stretch, and even though we're still stretched, not quite as much activity. And then when we're not stretching, whatever the opposite of stretching is, like I said, I'm not good with words. Once we release and allow that muscle to compress, no activity. So you see the biggest change while we're changing muscle length. That's where we have the biggest response for our type 1A. Type 2 basically just matches the length of the muscle. When it's stretched, I got my greatest firing rate. It kind of ramps up as we're stretching, and it kind of slows down as we're releasing the tension. It doesn't adapt. So it'll maintain that firing rate so long as we maintain the stretch on the muscle. The Golgi tendon organ afferents, they adapt a little bit, but not a whole lot. But the thing that they really sense here is tension on the ligament. So what we're doing here, we're pulling on two ends of a muscle. That doesn't put a whole lot of tension on the muscle. But if we apply tension and then the muscle contracts with that same tension there, that's what really stimulates our type 1B afferents. So 1B, kind of fast, right? Faster than type 2. But they're not rapid adapting like 1A. And the thing that they're going to sense is tension at the tendon. So in this cartoon, what they're doing is applying a passive stretch. And rather than releasing the muscle so that it can shorten, they stimulate it to contract. So this is stretch. Instead of letting go of the stretch, we contract. So we're adding even more tension in that case. So you'll notice over there on the right, while there's muscle contraction, the type 1B fibers, that's when they really come online. So not a whole lot here because they haven't put any tension on the muscle. When they stretch the muscle, you get some activation of the Golgi tendon organ, and then when the muscle is electrically stimulated to contract while still being stretched, that's when you really see the Golgi tendon organ activated. And these are going to create different reflexes. If we sense stretch of the muscle with our 1A afferents, that stimulates the lower motor neuron to cause contraction. If we sense a very high amount of tension with our 1B afferents, then we inhibit the motor neuron to try to protect the connective tissue. This is why you will have folks contract their muscles a whole lot in order to improve the stretch. So you're trying to stimulate the Golgi tendon organ by having them apply a whole lot of pressure onto the muscle. So having it contract even harder and harder without actually changing its length. So by holding the position still and applying force, because the muscle is not allowed to actually change its length, it's still pulling on that tendon. And so the more force you generate, The more you pull on the tendon, the more you stimulate 1B afferents. And thus the more you inhibit motor neurons, so the muscle relaxes a whole lot more. After that, isometric contraction, that might be the term, I don't know. Don't quote me on that one. Alright, so this is introducing what we already know. There's a difference between extrafusal and intrafusal fibers. So the extrafusal fibers, that's the big chunk. If you go by a steak, you're looking at extrafusal fibers and some fat. Intrafusal fibers flank the spindle. So we got our big alpha lower motor neurons for the extrafusal. Of course, there's still type S, FR, and FF here. We're dealing with extrafusal fibers with the motor units. The intrafusal fibers have to do with changing the length of the muscle, because there are reflexes built in to prevent changes in muscle length, involuntary changes in muscle length. So there's a bit of a reflex. I'm going to get rid of this and draw that out for you. All right. So if we stretch a muscle, maybe by tapping a tendon or something like that, putting pressure on that tendon is going to pull on the muscle. Now, muscle stretch is, of course, detected by 1A afferents at the muscle spindle. So they're in here somewhere. They get stimulated. This drives a reflex in the spinal cord. So let me get one of those up here. Let's see. The 1A afferents live, of course, in the dorsal root ganglion. Got to get that up. All right. These create a reflex with motor neurons. So when the motor neuron is active, that causes contraction of the muscle. So we go back to the original length. Imagine that that is the same as that. So stretch causes contraction. And you've probably noticed, if you tap a tendon, you stretch a muscle, it causes contraction. So you see a leg kick or something like that. So nice little simple reflex built in. And this is the stretch reflex. This is what prevents involuntary muscle movements. Because our muscles, they're going to change length just a little bit. I mean, you know, we're going to sway a little bit with gravity. So to keep us upright, anytime our leg muscles change their length, the stretch reflex kicks in. If the muscle stretches, that stimulates 1A afferents, so they stimulate the motor neuron to cause contraction. If the muscle shortens a bit involuntarily, that decreases 1A afferent activity, decreases muscle tone, and allows the muscle to lengthen a bit. So, kind of nifty. The stretch reflex is there to resist changes in muscle length. So we could stretch by tapping a tendon. Alternatively, we could tell a little old white lie. We could act at the level of the muscle spindle. Now remember, we have intrafusal fibers flanking our spindle. So what would happen if a gamma lower motor neuron, a little bit smaller, were to stimulate the intrafusal fibers? Well, they're going to contract. So when our intrafusal fibers shorten their length, that stretches the muscle spindle. So gamma lower motor neurons are recruited earlier than alpha lower motor neurons because they're smaller. V still equals IR, just like it did a few slides ago. Lower motor neurons first stretch the muscle spindle. We still have our spindle afferents around here. So the 1A afferents sense that, provide a little additional, sorry, I have to go in this way, a little additional excitatory input to the alpha lower motor neuron to help drive contraction. So when the muscle contracts, that squeezes everything together, so my spindle comes back to the same size, at least the sensory section of it, and my interfusal fibers stay contracted. So that I'm now where I started from, but my whole muscle has shortened, in length. It starts with the interfusal fibers. First, gamma lower motor neurons contract interfusal fibers to stretch the spindle. That helps drive activation of alpha lower motor neurons that act on extrafusal fibers to contract the whole muscle. The muscle changes its length, so it shortens. That also shortens the muscle spindle to go back where it was. So the gamma lower motor neurons determine the set point for the muscle. The more active our gamma lower motor neurons are, the shorter we're going to allow our muscle to be, because we trick the muscle spindle. We have to do that. The reason why this all works is because gamma lower motor neurons are much smaller than alpha lower motor neurons. There they are right there. Look on the bottom left. That's a little tiny gamma lower motor neuron, and on the right you can see a much larger alpha motor neuron. V equals IR. So the cell on the left has a much higher resistance and therefore is going to hit threshold potential sooner than the big motor neuron. This way we can trick our spindle into making it think that the muscle is longer than it should be, and that's going to allow contraction to occur. If we didn't do that, when we contracted, we'd unload our spindle and we'd stimulate a stretch reflex that would cancel out that contraction. So, nice little system here, and this is showing you the circuit that I just drew out for you. Starts with gamma lower motor neurons in the spindle. The sensory components of the spindle help drive the alpha lower motor neuron, and that acts on the extrafusal fibers. Here we can see what would happen if we only had alpha lower motor neurons. So on the left, that's kind of normal, or just some stretch, doesn't matter, we're not trying to contract. In the middle, if we contract with only alpha lower motor neurons, and we don't stretch our spindle first, the contraction of the whole muscle is going to shorten the length of the spindle. So the 1A afferents are going to have a drop in their activity. They won't provide any excitation to the alpha lower motor neuron, so it will be difficult for the alpha lower motor neuron to continue firing. On the other hand, if we want to maintain that shortened muscle length, we need the alpha lower motor neuron to continue firing. So what we do is have alpha and gamma together. Gamma starts a little bit earlier because it's smaller, stretches the spindle, everything I just said happens there. So stretch the spindle, contract the muscle, and go right back where you were. Maybe this will help. Here's my muscle spindle. My fingers are interfusal fibers, and my elbows here are the extrafusal fibers. So I want to make sure my mask doesn't droop down. Right, this is my set point. This is what the muscle wants. It wants the muscle spindle to stay this length. So if it ever changes, well, that tells me that my muscle has changed length and it shouldn't have, so fix it. So if it's stretched, decrease the muscle length, contract. Right? And if it's unloaded, well, relax the muscle. Maybe it had a little bit of involuntary activity. So, intrafusal fibers, extrafusal fibers. If I just contract extrafusal fibers, I'm going to unload my spindle. But if I contract my intrafusal fibers to stretch it first and then contract my extrafusal fibers, sorry, I dropped that, now I can maintain tension on the muscle spindle. So as far as the spindle is concerned, nothing has happened. We haven't actually changed muscle length. So as long as these two neurons are acting together, the alpha and the gamma, we can have a change in muscle length. If they're not acting together, then the change in muscle length is determined to be an error, and the stretch reflex helps offset it. Outside the muscles, we also have sensory components. And really, they're going to sense the same thing for the most part, and that is stretch. That's the big thing that we pick up, is stretch. Changes in pH and change in temperature are interpreted as pain. Everything else, stretch. Now, the body is not innervated equally. Some areas are innervated more densely, like our fingertips, so we have very fine sensation there. In other areas, like our back, for example, not so much. And an easy way of measuring the density of innervation is the 2-point discrimination test. And that's what's shown here. So on the x-axis, that's just how far apart they had to spread 2 points in order for it to be perceived as 2 points. If a part of the body receives fewer neurons, fewer sensory neurons, then each sensory neuron has to innervate a larger area of skin. And that area of skin innervated by a neuron is just called its receptive field. That's just the territory for any sensory neuron. If it's a visual neuron, then it's which part of the retina, or which part of the visual field does it pick up. But since we're talking about somatosensation, it's what patch of skin have we innervated. So with low density innervation, then each neuron, let's say we only have 2 neurons to cover this patch of skin. Well, this one's going to innervate a fairly large area, and this one will innervate another fairly large area. Same patch of skin, but we increase the density of innervation. Each neuron covers a smaller territory, so it has a smaller receptive field. We can only tell 2 points apart from one another if we stimulate 2 different neurons. So if I were to take some calipers and spread them apart, about this much, put them on here, whether it's 1 point or 2 points, I'm only stimulating this neuron, it's perceived as 1 point. Higher density, stimulate 2 neurons, perceive it as 2 points. You can put the calipers fairly close together when we're talking about the fingertips, the lips for sure, tongue, but whenever we get to the back, your thighs, I mean, if you're feeling around for your keys at night, you don't rub your back over the table, right? You're going to send your hands out. They can stretch further, and then they have much finer sensation. So we have different degrees of innervation depending on which area of the body. We don't innervate all parts of the body equally. And that's what accounts for that different sizes in the homunculus. If you look at a cartoon representation of the homunculus, they got massive lips, big hands, large genitals as well, and that's because the number of sensory neurons dedicated to those body areas is much higher than other areas. And as such, they occupy a greater surface area or volume of our cortex. And as I already mentioned, some afferents are adapting. Some aren't. So whenever you apply pressure, some sensory axons are going to fire action potentials pretty much the whole time. That'd be slowly adapting, shown in black there. Others are more responsive only at the beginning and only at the end. So when you apply the pressure or when you take the pressure off. So they're more sensitive to changes. This is very similar to what we just went over with the muscle spindles. So type 1A, rapid adapting, that'd be kind of like the blue there. And then the type 2, slowly or perhaps non-adapting, that's more like the black there. In case you care, the reason that we have adaptation is because of calcium-gated potassium channels. So as we continue to fire action potentials and bring in calcium into the neuron, that calcium turns on potassium channels, bringing us closer to potassium's reversal, which is, of course, below threshold, somewhere in the ballpark of -100. If you don't care, that's cool too. You just need to know that some fibers adapt. Now these fibers, if we're talking about tactile sensation, aren't just bare nerve endings sticking in the skin. They associate with specialized receptors. And there's 4 types, kind of in 2 locations. You could think more near the epidermis. That'd be the ones that begin with M. And then there's the ones more in the dermal layers. Those would be the ones that don't begin with M. So our Meissner corpuscles and Merkel's discs, a little closer to the surface. The corpuscles, the Pacinian corpuscle and the Ruffini corpuscles, they don't begin with M, so they're not at the surface. Maybe that'll help you remember it. The Merkel's disc and Ruffini endings that kind of have more of an R sound, they're not rapid adapting. You might expect them to because of the R, but no, it's the opposite of what you think, because of course it is. The rapid adapting ones don't have a strong R. I know Meissner has it in there, but work with me, okay? Pacinian at least doesn't have an R in it. Maybe that'll help you remember. I don't know. But the location and the degree of adaptation tells you about the information that they transmit. So for example, the Meissner corpuscle. You know, kind of near the surface, right? Pretty close to the epidermis there. Rapid adapting. Because it's rapid adapting, it needs to have on, off, on, off. So we're dealing with vibration. But it can be fairly weak, fairly low frequency vibration. So if something moves across the skin, that kind of vibrates the skin, Meissner corpuscles will pick that up. But it really only senses change, so it's more sensitive to movement along the skin because it's rapid adapting. Also rapid adapting would be the Pacinian corpuscle, but they're a little deeper, so you need something a bit stronger, not just a little movement on the skin. You need real vibration, like, you know, power tools or something like that. Those that are slow adapting, so the Merkel's discs and the Ruffini corpuscles, those slow adapting fibers are going to sense the either gentle deformation, so like reading Braille or something, that'd be a Merkel's disc, right? Because it's near the surface and it's slow adapting. So you can sense the shape of things. Like I can tell that there's, you know, these little grooves on the dry eraser marker. I don't need to go deep for that. And then the Ruffini corpuscles, those are going to sense, you know, a little more pressure. These are certainly going to work together. But things like if there's stretch on the skin, you know, because maybe you're holding something heavy and that kind of pulls on the skin a bit, the Ruffini corpuscles will pick that up. And they can pick that up because they're slowly adapting. They just need a little stronger input, I suppose, or a little deeper input, I should say, than the Merkel's discs. So knowing the location and knowing the pattern of adaptation, that tells you the type of information that's transmitted. No matter what, though, it all comes down to stretch. These are mechanically gated ion channels. And everything is to some degree, but these are strongly mechanically gated. So they're very sensitive to little tiny stretches. Of course, you can rub your eyes and you'll see colors because you're forcing open ion channels. Everything's mechanically gated to some degree. These ion channels, however, are attached to the cytoskeleton in such a way that makes it very easy for them to be pulled with a little bit of stretch. And so you can see it over here in the cartoon. There's the closed on the left, open on the right. So when there's stretch of the skin, it physically pulls open ion channels in the axon to initiate action potentials. And then there's a cartoon on the left just showing you the location of those different somatosensory receptors. So you'll see some kind of closer to the top, right, the ones that begin with M, and the ones that don't are down below. And just remember that kind of harder R sound. Those are slowly adapting. Now we also innervate the hair, of course. Now, unlike the other fibers that innervate the skin, that deal with non-painful stimulation, those are A beta fibers. A little bit bigger, type 2, same thing, same size anyway. In the hair, we're using A delta fibers, which are normally reserved for pain, but here's a little exception for you. And of course, you can get some pain if you pull on your hair, but the A delta fibers are a little bit smaller. So we got, and of course this is in the table, as far as size goes, alpha, bigger than beta, gamma, then we got delta, smaller. smaller it is, the higher the resistance, the more excitable it is. So little tiny changes in hair, you can feel the wind in your hair a little bit. Hair is a little more sensitive as a result because it has a smaller fiber that innervates it. Minor point. But our A delta fibers, if you're thinking A delta, it's totally fair for you to think pain and temperature. That's a big thing that they sense. And I guess to finish this off, we got C fibers at the very bottom. Those are small. So A delta and C fibers are there for pain and temperature sensation. A delta, myelinated, so it's quite a bit faster than the C fibers. C fibers are unmyelinated, they're very small, so it makes them easily excited, but they're very slow to tell you about it. So we have kind of two waves of pain, the A delta fibers, which kind of create a little sharper pain, as opposed to kind of a burning or itching pain that C fibers generate there. But they kind of serve different purposes. A delta is there to tell you somewhat quickly that you have pain and where it is. And then the C fibers are going to provide sort of a constant reminder that you have pain. So don't use that area again, not until it's healed up. That's because of, in part, the axon reflex, which we'll hit right at the end, but we did hit it last semester, so you already know what I'm going to tell you. So what is pain? Right, what are we sensing? Well, for the most part, acid. We're sensing changes in pH. There's kind of two things we're using here, not mechanically gated ion channels. Instead, we're using acid sensing ion channels, and these are mostly sodium channels, but they'll move some other cations. They're excitatory. They'll sense acid. If there's a drop in pH, they're stimulated. Trip channels. These are non-specific cation channels that are sensitive to changes in pH and heat. So there's where the temperature comes in. Some are stimulated by heat, some are stimulated by cold. So cold can hurt, hot can hurt. So really the trip channels, the transient receptor potential, there's a bunch of these. Some respond to hot, some respond to cold. They're all going to respond to pH to some degree. They're also stimulated by capsaicin. That's what you find in spicy foods. That's why spicy food hurts, because it stimulates pain receptors. So it actually hurts. Now there are other proteins on our pain sensing fibers that affect the conductance of these two ion channels, acid sensing ion channels and trip channels. So if we have tissue damage and things like ATP and bradykinin have leaked out, those stimulate G protein coupled receptors that potentiate activity through our trip channels. So the basic mechanism here is we have an ion channel. Here's our membrane. We've got an ion channel, we'll just say trip channels here, that are going to sense a change in pH or temp. But how sensitive we are to those changes in pH or temperature really depends on what else is going on. So if there's tissue damage, well, we're going to be a little more sensitive, right? Because intracellular components like ATP or bradykinin leak out, and they stimulate G protein coupled receptors. These are GQ, mind you. So we all know that stimulates phospholipase C. That's going to cut up PIP2, right? That's going to make a little diacylglycerol and IP3. Eventually, that's going to cause some calcium release. You remember that. Stimulates a protein kinase called protein kinase C, and that phosphorylates our trip channels to increase their conductance. So don't worry about all this junk. What this translates to is an increase in conductance through our trip channels. So they generate much stronger currents with the same change in pH or temperature. So temperatures that didn't used to hurt can now hurt, right? Even a little bit of pressure might hurt as a result. So the nociceptive afferents become more excitable because of metabotropic receptors. But the primary sensation of pain is through ion channels. Predominantly, they sense changes in pH. And that's why working out hurts. So early on, the drop in pH that occurs following exercise stimulates nociceptive afferents. So you get a little bit of early burn. Right? And the table over here, and there's a citation you can check out. The table over here just shows you that after exercise, there's a noticeable drop in pH. So if you just look on the bottom. The drop in pH, of course, means an increase in acid, and that's what these sense. So the reason that muscles burn or start to hurt after exercise is because of stimulation of trip channels on nociceptive afferents. Afterward, so the delayed onset muscle soreness, that's most likely due to inflammation. Inflammatory cytokines can also stimulate receptors to increase activity of trip channels. So if we have any damage to the muscle, you worked really hard, of course, damage causes inflammation. Inflammation requires an increase in blood flow to the site. So you'll get an increase in blood flow, you'll get a little bit of fluid pulled out of the blood. This creates kind of little cesspools of gas exchange, if you will, so it's easier to have a drop in pH. Guess what? That's of course going to stimulate trip channels. And so those damaged muscles are going to feel sore. You're going to feel pain as a result because of inflammation. Inflammation causing a drop in pH, and inflammatory cytokines causing an increase in conductance of our trip channels through metabotropic receptors, both of which lead to pain. Now the pain-sensing fibers also lead to inflammation themselves. Don't forget the axon reflex. This thing is still true. So when we sense pain in an area, it's probably because there's damage. So we should hopefully stimulate some inflammation as a result. So when my pain-sensing fiber here is very active, it releases neuropeptides like substance P, right? Calcitonin gene-related peptide, CGRP. These stimulate mast cells. The mast cells release histamine. Histamine stimulates the nociceptive afferent, so that area that was damaged and is in pain continues to be in pain. So we don't just get a little damage, or we don't get a little pain just when the damage occurs. We get continued feelings of pain even after the damage has occurred, until the healing process is complete. And near the end, once it's kind of scabbed over, let's say if you have a cut or something, you get a little itchy around that area, and that's because of low levels of C fiber activation because of the axon reflex. So that little bit of inflammation that's still sticking around stimulates C fibers at a very low level. But C fibers, because they're so small, are very excitable. So it's easy to stimulate them because of Ohm's law. So a very low level of inflammation leads to a low level of C fiber activation, and we interpret that as itch. And that, I think, covers it. I'm sure you're itching to ask me some questions, so put it in the questions box. We can go through it in class. See you later.